Hello students, welcome back to our biology lessons. We had started the biomolecules in the previous lesson and we had covered carbohydrates and lipids. In carbohydrates we had seen the sources, the structure, the function and the test of carbohydrates. In lipids also we had seen that basic sources, structure, function and the test. How can we test the lipids? So today's objective is we'll start proteins, which is the third biomolecule we'll learn, and in proteins we'll see how it looks, the basic structure, the list of various amino acids, the types of amino acids, the classification of proteins, and finally the functions of proteins. So let's zoom in.
Did you think what has made the structure of your body? It is made up of proteins. You may have studied in your lower standards. The proteins are the building blocks of every living organisms, which means that whatever structure the living organisms has is composed of proteins. But from where this protein comes from within our body? These come from mainly the food that we have consumed before or we are consuming in our diet. Our body has disintegrated these proteins which were present in the food and has broken down into small components. We call them as amino acids. So amino acids are the building blocks of proteins. And every two amino acids are joined together through a peptide bond. So whatever food or protein food we eat, they are disintegrated or broken down with the help of the enzymes which are present in our stomach and small intestine. And then we get a lot of building blocks or small components. We call them as amino acids. So as I told that two or more amino acids are joined together by peptide bonds. And a polypeptide chain of many amino acids is formed. So you can imagine this polypeptide chain like this necklace. You have seen that these pearls, the two or more pearls are joined together. So we can imagine this each pearl as a building block or the amino acid. And they are forming a long chain of peptide bonds. We call them as polypeptide. Now please remember, this polypeptide chain is not a protein. A polypeptide chain, when folds to form a stable structure, function it very efficiently and then they form the proteins. So this proteins, because within the proteins after folding, there are a lot of bonds formed like ionic bond, hydrogen bonds, etc. And then they form a very stable functional unit. Now let me discuss the elements present in amino acids. So the main elements present in amino acids are carbon, hydrogen, oxygen and nitrogen. Some amino acids have also sulphur and other elements, but mainly which must be present within the amino acids are carbon, hydrogen, oxygen and nitrogen. Now this is the basic structure of amino acids. So amino acids are organic compounds. Organic means what, students? Organic means any compound which has carbon within it. And amino acid is also organic. Why? Because it has a carbon within it. Okay. So these are organic compounds containing amino group. Now the NH2 is a functional group which is named as amino group. And this C double bond OH is a carboxyl group. Now please remember that this NH2 group is basic in nature, whereas this carboxyl group is acidic in nature. And the whole amino acids has NH2 as functional group and carboxylic acid as another functional group. Now, if you can see here that all the elements that are attached to this center carbon in an amino acid are different. One is NH2, one is hydrogen, one is carboxylic acid, and this are, so you know that carbon has a valency of 4. So 4 bonds can be formed with a single carbon atom. So 4 bonds, 4 different types of bonds that are formed are with amino group, with carboxyl group, with hydrogen group. This is not changing, never. So what is changing only is what differentiates from one amino acid to the other is the R group. Okay. So now if you see that these are 4 different groups, right, with the center carbon, and this type of, if you see any carbon which has 4 different groups attached to it, that means it has a very special property in chemistry. We call them as chiral carbon. It is a property, and what is that property? It makes this compound optically active. Okay. So this chiral carbon is present in every amino acid. Think about that. Pause the video and think about that. Does every amino acid is optically active, or does every amino acid will have a chiral carbon? Pause the video and think about that. Now if you see here, if the R group is replaced with hydrogen atom, that means the two groups have become the same, right? And hence it loses the optical activity. It is not chiral. And this particular and only amino acid which is not optically active is glycine when the R group is hydrogen. So please remember the basic structure of amino acids. That means whenever a carbon is bonded with amino group, carboxyl group, hydrogen, and any other R group, it can be hydrogen or any other group, then we call them as amino acids. Now, how does the two amino acids combine together? Let's see that. So if you take two amino acids, for example, this one is the first amino acid, it has an amino group, it has a carboxyl group, it has hydrogen, and it has any R group. And we take a second amino acid, if you can notice a similar structure, like this is the amino group, this is the carboxyl group, this is the R group, and this is the hydrogen group. So we bring the two amino acids together, then think what can happen. So you can see the blue letters, these form the water. So whatever water, if we remove from these two amino acids, then we can see that this carbon and this nitrogen, so carbon of one amino acid and nitrogen of the second amino acid can join together, just like this diagram. And this bond of carbon nitrogen, we call it as peptide bond. So students, it should be very clear in your mind how the peptide bond is formed. The peptide bond is formed after removal of one water molecule, right, from the two amino acids, and the carboxylic carbon and amino nitrogen join together to form the peptide bond. If you can see in this diagram also, the amino group and the carboxylic group, the water is removed and peptide bond is formed. Similarly, in a polypeptide chain, a lot of peptide bond can form, correct? So they form a heteropolymers. What does hetero mean? Hetero means different. Why they are different? Because you can see the R group. So what differentiates one amino acid from the other is the R group, this A and B group, designated with the letters. So these are heteropolymers. So proteins are a chain of heteropolymers of amino acids. It is not necessarily that all the R groups will be same. They are different. Okay. But what is same is the amino group, is the carboxylic group, and the hydrogen. Now, if two amino acids, this example is taken of glycine, but you remember that this hydrogen can be different. So, for example, if two amino acids join together and remove one water molecule, they form the dipeptide. So, two amino acids form a dipeptide. Similarly, if you take three amino acids together, so they form a tripeptide. And this CN bond is called the peptide bond. What CN bond? The carboxylic carbon of one amino acid and amino nitrogen form together a peptide bond. Similarly, when a lot of peptide bonds are formed, we call them as polypeptide. Let's see the list of amino acids. Students, there are 20 different amino acids present within our body. They are glycine, alanine, serine, tryptophan and proline. So these 20 amino acids. At present, don't get nervous. It is not necessary to memorize all these amino acids. But yes, for the future, you need to understand the functions because all these amino acids have different properties. Okay. Now, among these all 20 amino acids, these all amino acids are not essential. What is essential amino acids? Which need to be taken through our diet. They cannot be synthesized within our body. So, they are essential. We need to take it. We need to take it in the form of a food. But what are non-essential? Non-essential amino acids is just the opposite of essential. That means our body can synthesize the non-essential amino acids. We may not consume them, but what we should consume and must consume are the essential amino acids. Now, I will show you, this one is important. I will show you a list of essential amino acids which are necessary for our body. They are histidine, isoleucine, leucine, methionine, phenylalanine, threonine, tryptophan, valine, lysine. Now, how will you memorize it? This is a mnemonics that you can write in your notebook. This is the mnemonics, that is private Tim Hall, which means P for phenylalanine, V for valine, T for tryptophan, private, threonine, T, isoleucine I, methionine M, Tim, and Hall, that is histidine, arginine, leucine, lysine. Okay. These are the essential amino acids. Students, these amino acids need to be consumed through the diet. Okay. These cannot be synthesized by our body. Now, these two amino acids, you see that they are called semi-essential, histidine and arginine. Why? Because whatever quantity is required for our body, that cannot be substituted through our body. So, our body makes the histidine and arginine, but not in sufficient quantity. So, they need to be consumed through our diet. So, they are called semi-essential. So, if in your question, students, it is asked, what are semi-essential? That means you can say that semi-essential amino acids are those amino acids which are produced by our body but not in sufficient quantity. They need to be substituted from the diet. Okay. Students, let's learn about the types of amino acids. So, if you see, it's a very important one and it has come many times in your examinations. So, please pay attention here. This form, we call it as zwitterionic form. You can see the picture in the center. That is, it has both the positive charge and the negative charge. You can see that there is an extra positive charge, extra hydrogen ion attached to the amino group of the amino acid. Similarly, the carboxylic group has a one hydrogen less and hence it has a negative charge. Hence the total charge, it is 0. You can see here this zwitterionic form It is a form of any amino acid where if it exists in this form, we call it as zwitterionic form. Now this is a pH scale. You can see it is increasing pH. So low pH means it is acidic one, and the higher pH means it is basic one. So starting from the neutral pH, you can see here that here the net charge is 0, where it can exist in the zwitterionic form. And neutral pH we also call it as Pi, or the isoelectric point. You will learn more details about it in your chemistry lesson. Now coming to the form of amino acid in lower pH or in acidic solution. In acidic solution, every amino acid can gain a positive charge in the amino group, okay, and hence the pH is acidic. But what will be the nature of the amino acid? It will be basic. Similarly, if you see here in the higher pH, the net charge will be negative, the pH is the basic, and the nature of amino acid will be acidic. Okay, please understand this diagram carefully and note it down in your notebooks. Now coming to this one again, that in lower pH we can see that since there is one hydrogen ion, it can donate hydrogen ions in solution. Similarly, in the neutral basic pH, they can accept hydrogen ions in solutions because they have a negative charge, and in neutral form the net charge is 0. Now my question to you is, does the structure of amino acid changes in different pH solutions? Think carefully about this question. Does the structure of amino acid changes in different pH solutions? Pause the video, provide the answer. Yes, the structure of amino acid changes in different pH solution. Just now we have seen there are 3 forms of amino acid that can exist in different pH. One is the acidic one is the zwitterionic form, and one is the basic one. Okay. Now coming to, I will only show you only 3 types of amino acid. Here we call it as aspartic acid. This is the structure of aspartic acid, and if you can see, this is the amino group, carboxyl group, hydrogen, and the R group. The R group has CH2, COOH. So there is one extra carboxylic acid group. So this is carboxylic acid group which is attached to the chiral carbon. That is different. But there is in the R group also, there is a carboxylic acid group. So whenever the word acid is there, that means it is acidic. So this amino acid is an example of acidic amino acid. Similarly, this one, if you can see, the R group is hydrogen. Even if the R group is something like aromatic or alcoholic like serine, so it is neutral. This is an example of glycine. Here it is neutral. Now if you can see the third one, here the R group is a very long chain like CH2, 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 and there is an extra amino group NH2. So one apart from one amino group, there is a second amino group in the R group, R chain. So this example is of lysine, and this is basic in nature. So we have learned there are 3 types of amino acids depending on the R group. Okay, so if the R group varies, the nature of the amino acid also varies. But the amino acid exists in different forms in different pH solution. Important thing to remember, this lysine amino acid plays a very important role in our body. If you know, give me the answer in the comment section. So classification of proteins, we can divide proteins into 2 parts, simple proteins and conjugate proteins. In simple protein, one example is histone, and histone plays a very important role in our DNA, right? And this is composed of amino acid and its derivatives. What does it mean? That if you hydrolyze this type of protein, if you break down to this type of protein, you will only get amino acids. Okay, nothing else. So you will get only amino acids. But what happens to conjugate proteins? Conjugate proteins, if you break down into parts, then you will get not only amino acids but also another part, we call it as prosthetic group. So here in this case you can see that there are two types of conjugate proteins. There are more than two types, but for now you can learn only two types of conjugate proteins. One is the lipoprotein. Lipoprotein from the word, so segregate the word. You can understand that lipo stands for lipids and proteins. So it has a prosthetic groups of lipids, okay, apart from proteins. And here in glycoprotein, you can guess that it is carbohydrate and protein. So these are the conjugate proteins, and this one is a simple protein where you can get only amino acid and its derivatives. Now coming to the functions. Functions, so proteins are a very important part of our life. We can see different roles of protein. For example, the first part is the keratin, which acts as a structural protein, and keratin is present in our hairs. Okay. The second part, it can also play a very important role as an enzyme. For example, trypsin or amylase. Okay, trypsin is present in our small intestine and amylase is present in our mouth or saliva. So they can play a very important role as enzymes. They can also play a very important role as messengers. For example, hormones, and insulin is the best example, where insulin plays a very important role in maintaining our sugar level in our body, right? Similarly, proteins can also fight very infectious disease and by creating antibodies within our body. So you see how proteins are important, and this one is very important function. Like you can guess, it is a hemoglobin, and it acts as a transport protein. And you know that already that hemoglobin plays an important part in transporting oxygen to various parts of the body from the lungs. Similarly, the myosin you can see, which is present in muscle cell and is a contractile protein. So, to conclude, we have seen the basic structure of the amino acids, which has an amino group, a carboxyl group, a hydrogen group and an R group. R group is different for different amino acids. Similarly, we have seen how the peptide bond is formed by removing of a water molecule. Also, we have seen that the proteins are the heteropolymers of amino acids and have a polypeptide chain. It has a lot of peptide chains within a protein molecules, and polypeptide chain is not enough. The folding of the polypeptide chain makes the protein functional. Similarly, we have also covered that there are 20 amino acids, out of which 10 are essential amino acids which our body cannot make or cannot produce, and they need to be consumed through our diet. Also, we have seen that the types of amino acids, that is the acidic, neutral or basic. Acidic means which has more than one carboxyl group in the R group. Neutral means the number of carboxyl group and the amino group is same. And basic means the R group of the amino acid contains an extra amino group, which makes the structure or makes the function of that particular amino acid basic in nature. We have also seen that the classification of proteins as simple proteins and conjugate proteins. Simple proteins are the proteins which when hydrolyzed gives proteins and its derivatives, whereas conjugate proteins can be of two types. Mainly it is lipoprotein and glycoprotein. Lipoproteins is lipids and proteins, and glycoproteins is carbohydrates and proteins. Finally, we have seen the various functions of proteins, which acts a major role in transportation, as they act as hormones or messenger molecules, enzymes, antibodies, and structurally like the keratin.
So students, we have come to an end of our today's lesson. I hope that you have understood proteins, biomolecules and its functions. In the next class, we will start nucleic acids, But before proceeding further, don't forget to write your comments about the challenged question, which is just after this. Thank you for watching.